So, hello everybody. My name is Hanno Würbel, and I've been invited to give a presentation here on the issue of reproducibility in animal research. I'm a professor of animal welfare at the University of Bern in Switzerland, and I'm very happy to be here and to talk to you, even though it's a bit strange to talk to my notebook here, not seeing anybody sitting anywhere. But I hope this is going to be an entertaining presentation for you all. So I'm going to talk about biological variation and how that matters for reproducibility in animal research. And of course, reproducibility, or the reproducibility crisis, as it is called, has been a big issue over the last couple of years. And I'm going to particularly focus on the implications that has for animal research. And of course, if we're talking about poor reproducibility, then this does not only have scientific and economic implications, but in the case of animal research, it certainly also has ethical implications with respect to the use of animals. And as long as we're not guaranteeing that the use of animals is producing any meaningful knowledge, then of course we may risk wasting animals for inconclusive research. And that is certainly not in the interest of the population and society as a general. So the things or the sources of poor reproducibility that have been proposed, and these are not limited to animal research, but they also do matter for animal research, are several, and the most important ones I've listed on this slide. So there is a lack of scientific rigor, and that is something that has been revealed by meta-research, by meta-analysis, looking into papers and how they report methods and experimental conduct. And a lot of evidence suggests that in many cases, studies are not properly randomized. Scientists do not use blinding and do not use other measures of good research practice that are meant to avoid bias. And of course, if there is a risk of bias, there's also a risk of poor reproducibility. Then another issue, and that is particularly pertinent to animal research, is the use of small sample sizes. And of course, usually we do all we can in order to use as few animals as possible in order not to waste animals unnecessarily. But of course, if you're not using enough animals, then this may mean that you lack statistical power to detect the effect that you're looking for. And so in the end, your results may be meaningless simply because you have not used enough animals. Then another big issue is analytical flexibility in many cases, and that is certainly true for a lot of animal research. Studies contain or include subgroups of animals, different sexes, different age groups, different treatments, and often a number of different outcome variables are assessed. And so there is a lot of flexibility in statistical analysis. And of course, if you explore the data and look for statistically significant associations, and then present these as if they had been the result of a rigorously designed confirmatory study, then you may overblow your conclusions, and you may conclude things from spurious findings. And the final point I want to mention here is publication bias or selective reporting in general. So if several outcome measures are assessed, but then only the ones that turn out to be statistically significant are published, or whole studies are not published if they do not yield statistically significant results, then this is obviously going to distort the evidence that is published in the literature. And so we cannot rely on what has been published if a lot of results that did not show up as statistically significant have simply been put under the carpet. Now, I'm not going to talk about these things any further, but instead what I'm going to focus on is the issue of rigorous standardization. And this is something that is not usually considered as a source of poor reproducibility, but I'm trying to convince you in the course of this presentation that this is actually a major issue in animal research. Now, first, let's look into what rigorous standardization means. Standardization essentially means the defining of the characteristics of the animals and their environment, and to keep these characteristics constant. And of course, this leads to a homogeneous study sample that is studied under rigorously controlled and standardized conditions. And the important point I want to make here is that if we compare it to the other sources of poor reproducibility, they are all considered to be bad practice. They are actually violations of good research practice. But in terms of rigorous standardization, this is actually something that is considered as good practice. And many textbooks of laboratory animal science, they actually recommend to use rigorous standardization. And the intention behind this is, of course, to get results that are as precise as possible, and by reducing the variation in the results, to be able to use fewer animals for the same study. Now, if we want to understand why this could potentially be a problem for reproducibility, we first need to go back in history by about 350 years to the birth of reproducibility as a scientific principle to establish matters of fact. Back in the 17th century, the Royal Society at some point decided that in order to distinguish between anecdotes reported by scientists and robust scientific evidence, that they need some kind of quality control. And the measure that they took in order to do this was that they required from then on that in order for a result to be accepted as scientific evidence, it needed to be replicated by an independent laboratory. So at that time, in order to, for a result to be accepted as scientific evidence, it was not sufficient to just publish a single observation or a single study, but the study had to be replicated independently by an independent scientist. Now, of course, since that time, we've come a long way. And these days, as you all know, we don't need to plan multi-laboratory studies or independent replications in order to get funding. And we don't need to report independent replications in order to get our results published. Instead, what we're doing is, instead of just observing one animals, we build replicates into our study design. So the picture here illustrates animals waiting for a study, and instead of just observing one animal or one cage of animals, we have a sample size, a sample of animals that we're studying, and these animals are counting as independent replicates within our study design. Now, the problem with this is, of course, that the animals within such a study population, they are a lot less independent of each other than an independent observation in an independent laboratory by independent scientists. And in particular, if we come back to the rigorous standardization that is happening in laboratory animal science, these animals are usually identical, usually genetically identical. They may be inbred strains or even clones. They have the same age, they have been fed the same food, they are housed under the same conditions, and they are cared for by the same care staff, and in the end tested by the same experimenter. So they have lots of factors that shape their phenotype in common, and of course, the more these conditions are standardized within a laboratory, the more they risk to be different from the conditions in a different laboratory. And the first time that this got actually the attention of scientists or in the scientific community was a paper published in 1999 by 3 behavioral geneticists who got together to test different inbred strains of mice, the 8 strains that are listed here on this figure, and compared them in a number of different behavioral tests, as is usually done when new mutants are behaviorally characterized, or when these animals are used in order to screen for the effects of new drugs or new mutations. So as you can see on this picture, across the 3 laboratories, I've highlighted 2 strains in color. And as you can see on this particular outcome measure, an outcome measure of a conventional anxiety test, the elevated plus MACE test, you can see that the difference between these 2 strains in Albany goes in one direction. In Portland, it goes in the opposite direction. And in Edmonton, there is no difference at all between these 2 strains of mice. So I've put the term despite here in inverted commas, so poor reproducibility despite rigorous standardization, meaning that people were discussing, well, how could this happen? How can results be so different if, because these scientists, they tried to do as much as was possible to standardize the conditions, not just the test conditions, but also the housing conditions of these animals across the 3 laboratories. And yet they sometimes found completely different results between different strains. There were other cases. If we look at the 2 strains here on the very left, you can see that there is a little bit of variation between the different labs. But in general, the difference between the 2 strains goes in the same direction in all 3 labs, and there is a big difference between these 2 strains across all 3 laboratories. Now, the problem is that in order to know whether you have such a robust strain difference or a difference that may be spurious and may be limited to the specific conditions of your own laboratory, in order to know that, you need to vary conditions. It doesn't mean that you need to do a multi-laboratory study as here, but at least you need to introduce some variation in your study design in order to assess whether a difference is a robust effect or is very specific to the specific standardized conditions that were used in that experiment. And this is what I have referred to as the standardization fallacy, namely the attempt to increase reproducibility at the expense of external validity. And this is actually what happens when conditions are standardized within study. That is that there are, of course, many factors that can be standardized across laboratories: housing conditions, food, the type of animal being used. But there are also many factors that simply cannot be standardized. The microbiome of these animals will be laboratory specific. The personnel interacting with the animals cannot be standardized. And so there will be many environmental factors that cannot be standardized, but will still affect the animals in one way or another, and therefore produce different phenotypes between different laboratories. And of course, the more you standardize, the more rigorously you standardize the conditions within your study, the higher the risk that the results that you will obtain will be limited, or the validity of these results will be limited to these specific conditions. Now, I was by no means the first to mention this, and if we go back in history a little bit, you can see that Ronald Fisher, one of the godfathers of experimental design and statistics, mentioned this already back in the 30s of the last century, namely that standardization weakens rather than strengthens our ground for inferring a result. And this is because a standardized experiment can supply direct information only in respect to the narrow range of conditions achieved by standardization. So standardization reduces the inference space, the space to which we can draw inferences from the results that we have obtained. And then, of course, as is normally the case, when experiments get replicated in a different lab by independent scientists, the conditions are different, and therefore, if the results aren't robust, they will not be reproducible. And it is important when we're talking about animal research to understand that the variation in our studies are not just random noise, they're not just measurement error, but there is a lot of biological variation, and biological variation is the very essence of all living organisms. So animals differ by their genes. We know different strains of mice, they have different characteristics. They differ by sex. They differ depending on which environment they have been brought up in. So early experience, the rearing conditions, weaning age, all that can have a strong effect on the phenotype of the animals. The same applies to housing and husbandry conditions, and of course also the test conditions under which conditions the animals are tested will affect the outcome of a study. So what we're talking about here is gene by environment interactions, and of course every animal is the product of the interaction between its genes and its environmental history. And this leads us to the reproducibility paradox, and I've drawn a simple sketch here taken from a paper that we published a couple of years back, where we try to put the reproducibility of animal research into perspective with biological variation. So what I've drawn here is a so-called norm of reaction, a change in the reaction of the animals to a particular stimulus in response to a particular environmental parameter. This parameter X can be temperature, it can be noise, it can also be a treatment that is applied to the animals, and as you can see on the left side by the distribution of the trait values, depending on which level of the environmental parameter I select, I may get different results. Now we can translate this into replicate studies. So one lab does a study under its own specific standardized conditions and obtains a result, maybe the dark blue result. And then a different lab tries to replicate this study, but because the conditions in that lab are different, they will find a slightly different result, here represented by light blue. So this led us to suggest that if we want to obtain robust results that are actually generalizable across the conditions by which laboratories vary and therefore reproducible, we need to take this variation as it exists between laboratories into account. And so we predicted that if instead of a single laboratory study under highly standardized conditions, we would conduct a multi-laboratory condition that includes or incorporates all this variation between laboratories, we would obtain a less precise results as represented by the wider distribution of this blue distribution here, but the results should be more consistent and therefore better reproducible across different laboratories. And in order to study this, we were using real data, and this is an example of the data that we used. This is data of 222 studies on the effect of hypothermia on the infarct volume in animal models of stroke. Now, you may wonder, why was this experiment conducted 220 times? Of course, it was not all independent experiments, but because hypothermia is one of the very few treatments that are also effective in the clinical population against stroke or show an improvement in the outcome of stroke. Hypothermia is used as a positive control in many animal studies of stroke. So we had a lot of data to play with, and they are ranked here along the effect size that they produced. The gray bar indicates that on average, hypothermia reduces the infarct volume of a stroke by about 50%. So what we did with these data, we selected 50 of these 222 studies. We selected the 50 studies that were most independent of each other, meaning that they came from different labs and from different scientists. We then used the parameter estimates as they were reported in the papers, the mean and the standard deviation, in order to generate sample values. And with these data, we then ran simulations with different sample sizes. In some cases, we used sample sizes of 12, in others 24 or 48 animals per group. Samples were taken proportionally from either 1, 2, 3, or 4 studies in order to mimic single lab studies and also multi-lab studies, including 2, 3, or 4 laboratories. Now I'm going to show you this in an example here. So here you can see the design for a single laboratory study and for a multi-laboratory study. The multi-laboratory studies includes 3 laboratories. So in order to generate a multi-laboratory study, we randomly picked 3 papers from the stack of 50 studies, and then took the data as they were described in these papers, and then proportionally sampled from these 3 different papers in order to generate a multi-lab study comprising, in this case, 24 animals, so 12 each per treatment group, control and treatment, and compared this to a single laboratory study, in which case we randomly selected 1 study and then sampled all 24 animals from this particular single study. So here are results. These are 15 studies of single lab study simulations. So this is 15 independent studies taken from these 50 studies, and then data sampled from the distribution as described in these papers. And as you can see, the results vary quite considerably across these 15 studies. The red line is the null line, so meaning that there is no effect of hypothermia. And as you can see, 50 or 100%, the full spectrum and even negative results are represented in this sample of 15 studies within this selection of 50 studies that we looked at. The dotted line is the summary effect size across all 50 studies, so that is the roughly 50% reduction in infarct volume that you get when you do a meta-analysis across all 50 studies. We then looked for the proportion of studies that found a significant effect and an effect that accurately represents that summary effect. And by significant, we mean that the confidence interval doesn't overlap with the 0 line. And with accurate, we mean that the confidence interval of the study overlaps with the summary effect size. So as you can see from these 50 single lab studies, there were only 3 that yielded significant and accurate effect size estimates. Now, when we look at multi-lab simulations, you can see that already with simulations based on only 2 studies, we get much more consistent results, and this improves even further when we take 4 labs into account. So when we simulate the study across 4 different laboratories, as you can see on the right side of this slide. The number of results that are significant and accurate estimates of the overall effect size increases from 3 to 10 out of the 15 when we include 2 labs or 2 studies into the simulation. And if we include 4 labs, then on average we get 14 out of 15 of these multi-lab studies producing significant and accurate effect size estimates. And it is important to note that these multi-lab studies, they include exactly the same number of data points as the single lab studies. So it's not a matter of sample size. It's simply a matter of sampling across a wider distribution of data as it occurs when experiments are conducted across multiple labs. We replicated this analysis for 13 different interventions in animal models of stroke, myocardial infarction, and breast cancer. And in all cases, except for very small sample sizes in a few cases, we can see a similar pattern of improvement of the reproducibility or the consistency of the results with an increasing number of laboratories included in the multi-lab study. So whereas in most cases, single lab studies produced significant and accurate effect size estimates of in less than 50% of the cases, this number increased to 60 to 80% in multi-lab studies, including 3 or 4 laboratories. Now, one of the reasons that may explain this effect is the heterogeneity that exists between animal studies. Study heterogeneity here represented as I square over the 13 different data sets that we analyzed represents the proportion of variation that is accounted for by the laboratory or by the study rather than the treatment effect that was studied. And as you can see is that the large proportion, and in some cases almost 100%, and in one case even 100% of the variation in the data was accounted for by differences between studies rather than differences within studies, so differences between animals within studies. And this is a huge heterogeneity, which of course can be explained by the high degree of standardization within studies and the much lower degree of standardization between studies. And when we compare this with human clinical trials, you can see that this is a completely different pattern. Even though there are in human clinical trials, there are studies where you find large heterogeneities of 60 to 100%. The proportion of studies in which you find much lower values of heterogeneity is much higher in clinical trials. And we believe that one of the most important reasons for this is that, of course, the human clinical population is much more variable in each of the studies and is much less standardized within studies compared to different studies. And therefore, these clinical trials, they cover a much larger proportion of the variation that exists across different studies compared to animal studies, where we usually have a very narrow space, a very narrow inference space within studies and differences between studies. This is also shown by a recent study where scientists interested in the metabolic rate and the response to obesity in laboratory mice were analyzing a big data set across many different laboratories. They included nearly 10,000 black 6 mice, so all mice, inbred mice of the same strain. But what they found was that institution, so the laboratory where the study was conducted, had the strongest effect on the variation in the data. So again, an indication that differences between laboratories can be massive and may dominate the results of animal experiments. And if we don't take this into account, we may produce very precise but rather spurious findings that do not generalize even across the conditions that exist between different laboratories. So to sum this up, we need to be clear that standardized single lab studies produce highly unreliable results because they usually have a poor external validity due to the fact that the inference space is reduced by the high degree of standardization, and that therefore standardization can be an important cause of poor reproducibility. And what we propose is that instead of standardizing study populations, we should actively heterogenize study populations in order to improve the reproducibility. And this can be done without the need for more animals if we use advanced statistical designs, advanced experimental designs and statistical methods. So eliminating biological variation through standardization is a highly inefficient strategy for generating scientific evidence because it means that each specific genotype by environment interaction will be studied in a separate experiment, and this is akin to the atomization of animal research. And as such, each of those very specific studies will contribute very little to our information gain. So what are the solutions? Possible solutions, of course, we can go back as proposed by the Royal Society in the 17th century and do an independent replication. This can be a useful strategy, but of course, even though it is a simple strategy, it is a rather inefficient strategy if we think that every single experiment would have to be replicated independent in a different laboratory by other scientists. And there are certainly better solutions to this. One solution is multi-laboratory studies, as I've just shown you. Multi-laboratory studies are effective, but they are logistically highly demanding. It needs a lot of organization in order to bring different laboratories together and organize a study in a way that the conditions can be compared across laboratories. And so this is something that you don't want to do for every single experiment, but maybe a solution for a late-stage preclinical study before you decide to go into a clinical trial, perhaps. Then another possibility is simply to adjust the metrics of significance or the metrics that you apply in order to decide whether or not your treatment had an effect. And this is something that can be applied to research areas that have highly harmonized methods, as for example, the phenotyping of mouse mutants, where a lot of independent laboratories are doing similar types of studies using similar types of tests and similar animals. And in these cases, it is possible to simply adjust the p-values of single lab studies in a way that takes the random effect of labs into account. So if you have a good estimate of to what extent results vary across laboratories, you can take this into account statistically and adjust your single lab results by this factor. And finally, and this is the method that we are working on most, is instead of standardizing study populations, to actively heterogenize them in single lab studies. So instead of running a multi-lab study, to mimic a multi-lab study by producing sub-cohorts within a single study population, a sub-cohort of animals. This can be different strains of animals that are included, or it can be different housing conditions or different test conditions that are included in a study design. Here is an illustration. Heterogenization of study samples in single lab studies can either be done by systematically varying environmental factors, factors of housing conditions, husbandry protocols or test conditions, or by variation of genetic factors, so varying the species, the strain, or the sex of the animals. And randomized block designs are the perfect design that can be used for this. And in many animal experiments, a blocking design is appropriate anyway, because, for example, several animals are housed in a cage. And if you want to account for the dependence of animals that are housed within a cage, you would include cage as a blocking factor in the statistical design anyway. Now, once you do this and you have a blocking factor in your study design, there is no reason why you should not make these blocks more different between each other. And instead of having all the same animals in all cages, you may have one strain in one cage and a different strain in another cage. And some of the animals may be exposed to more stressful conditions, and other animals may be exposed to less stressful conditions. And by this, you create a range of genetic and environmental variation that may or should mimic the variation that normally exists between different laboratories, and through this get results that may be less precise, but will be more robust and have a higher likelihood to produce reproducible results than rigorously standardized study samples. So with this, I would like to thank here some of my collaborators who contributed to the data that I've shown here, in particular Bernhard Völkel, who contributed a lot both empirically as well as theoretically. And then my former PhD students, Lucille Vogt and Helene Richter, and my long-standing collaborator, Joseph Garner from Stanford, and other collaborators that have contributed to this work, in particular, Yoav Benjamini and Neri Kafkafi from Tel Aviv, and Emily Sena from Edinburgh. And on the right side, you see all the institutions that have supported me and my team with funding for this research. Thank you very much for your attention. And of course, I'm ready to answer questions. We cannot have a live chat here, but you're welcome to address your questions via email, and I'll do my best to answer all of them as quickly as possible.
Alrighty, great job. That is a wrap, so we're all set. And pretty much from here, I'll just do some quick editing on the front and back ends just to get it all ready for the event.
Yeah.
Alrighty.
Can I review this? I mean, can I see this once you've done it, or how do you proceed with this?
Definitely. After I'm done editing, I can send you a preview link and you can take a look at your presentation using that preview link.
Okay.
So I can definitely send that over to you once it's ready, and then we can go from there with any feedback you might have.
Okay. Thank you very much.
Alrighty. Any other questions for me before we wrap up here?
Too much?
Oh, no, you were totally fine. Yeah, okay. That was great. Yeah. Awesome. Well, that is everything. Do you have any other questions for me before we wrap up?
No, that's fine.
Okay. Sounds good. Well, have a good rest of your day, and I will talk to you soon.
Okay. Thank you very much.
Bye-bye.
Bye-bye.